Good morning. I'm pleased to present this morning a webinar on nitrosamines in medical products and how to assess the risks associated with that topic. This webinar is in 4 sections. This module 1 is an introduction to the topic and an assessment of the process risks associated with API manufacture. Module 2 will address other risks associated with API manufacture. Module 3 will address risks associated with the drug product. And in Module 4, there will be a brief discussion of testing procedures associated with nitrosamines. First of all, to introduce myself, my name is Turlough O'Creadon. I have been working in the pharmaceutical industry for more than 30 years, in total in healthcare manufacturing, including devices, for over 40 years. And I have been a qualified person since 1995, and I am working on this topic of nitrosamines through much of last year, 2019, and into this year. The background to this topic: in June 2018, it was notified to FDA that nitrosamines had been discovered in sartan products, and this was traced back to the API manufacturer at a company called Zhejiang Huahai Pharmaceutical Company, who are a major manufacturer of APIs. Throughout the rest of 2018, first of all, other manufacturers of sartans were found to be at risk. And then as time went on, through 2019, nitrosamines were found in other products. And this has led to a kind of crisis in the industry, with directives from EMA in terms of addressing the risks associated with that. In September of 2019, EMA issued an instruction to all manufacturers to assess the risks associated with the presence of nitrosamines in small molecule pharmaceutical products, and that risk assessment is due to be completed by March of this year, 2020. This webinar is intended to give some prompts and ideas in terms of carrying out that risk assessment, and at the end we will advise you as to where you can possibly get more information. So, as mentioned, there is a directive from the European Medicines Authority. There are similar directives from other authorities. The Canadians have also asked for formal risk assessment, as have the South Koreans and the Swiss. FDA has not asked for such an assessment, but FDA continues to be vigilant and is itself alert to the risks that are associated with this issue. So let's talk about the nitrosamines themselves. And the most common types, the 6 that are of most immediate concern, are first of all what is referred to as NDMA, or to give it its full name, N-nitrosodimethylamine. Secondly is NDEA, or N-nitrosodiethylamine, and NDBA, which is N-nitrosodibutylamine. And it must be noted that all of these are considered genotoxic and carcinogenic agents. There is significant toxicological information, particularly around NDMA, and in reality there is no level that is considered absolutely safe, but there are levels where the risks are considered equivalent to the normal environmental risks. It must be remembered that these compounds do exist in other areas besides in medical products. For example, they are commonly associated with certain types of cured meat and fish. Some of the other nitrosamines that are of interest, the first one, which I will refer to as BMSA, and it's also known as MNBA, is its full name is 4-methylnitrosaminobutyric acid. And then there are other types. There are two other types which are in the cohort of concern. These are DIPNA and EIPNA. So let's talk about how nitrosamines are formed. And the primary method of formation is by the interaction of secondary amines with nitrite ions. As you can see here in the diagram, the reaction forms a nitrosyl group at the amine. The R groups that are shown in the molecule can be the same or they can be different. So for example, in the case of NDMA, the two R groups would be methyl. In the case of NDEA, the two R groups would be ethyl groups. The European Medicines Authority, in one of their documents, has shown that nitrosamines can be formed by the degradation of tertiary amines in the presence of nitrous acid, that under the acidic conditions, there's a sequence of reaction which can form the nitrosamines even from tertiary amines. But this is a much lesser risk, and in practical terms, the risks of formation of nitrosamines from tertiary amines are much less significant than the risks of formation from secondary amines. Part of why this problem has gone undetected for so long is that it is not always clear that secondary amines are present in the API manufacturing process. However, the API manufacturing process can involve agents which give rise to secondary amines or contain secondary amines as an impurity. And one of the prime examples of this, one which is well known, is a material called dimethylformamide, and this is used in a number of API manufacturing processes. As shown here, the dimethylformamide can break down to give dimethylamine. The formic acid that is the other byproduct of that degradation can itself break down, so that dimethylamine is almost always an impurity present in dimethylformamide, and during the course of reactions to manufacture APIs can itself be formed, so that the risk of dimethylamine arising from dimethylformamide has to be considered. And if nitrite ions are present, then that dimethylamine will give rise to NDMA. Another compound, which is often not considered in terms of the risk of secondary amines, is a material called N-methylpyrrolidone. N-methylpyrrolidone is a 5-membered cyclic ring, and that ring contains an amide linkage that, again, depending on the reaction conditions, that ring can break down to give a butyric acid derivative with an amino group. And that secondary amine, in the course of API manufacture, can give you this nitrosamine molecule BMSA. So there are other sources of secondary amines. As I mentioned, it has to be considered not just the secondary amines themselves and whether secondary amines are present in the reaction, but secondary amines are often present as an impurity in tertiary or quaternary amines. So for example, if you are using a material such as triethylamine, which is commonly used in API manufacture, if you look at the specification for triethylamine, there will be there a limit on the presence of diethylamine as an impurity. Similarly, for dimethylamine, in the presence of trimethylamine. And as mentioned, simple amides such as DMF often contain the amine itself as an impurity. So when you put these risk factors together, the presence of secondary, tertiary, or even quaternary amines has to be considered a risk factor. And it must be remembered that the sources of secondary amines is not just the presence of the amines themselves, but they may come from other reagents. And indeed, it must be considered the risk of carryover. There may be a risk of secondary amines in one step, that if those are not removed in a subsequent step of API manufacture, they may be present as an impurity. The second part of the equation in terms of the presence of nitrosamines is sources of nitrite ions. Nitrite ions are sometimes used directly in reactions, in particular if the reaction uses sodium azide. Nitrite is commonly used to quench the reaction on completion, and that will give rise to the presence of an aqueous layer with the organic layer, and the separation of those two layers is critical to avoiding nitrosamines in the API. It must also be remembered that nitrites can be present as impurities in other reagents. So for example, I mentioned sodium azide, and it is common to have sodium nitrite as a minor impurity in sodium azide. So again, the specification for the sodium azide has to be reviewed, and it should be looked at what is the maximum level of sodium nitrite that might be present. A lesser source of nitrites are organic nitrites, and these can give rise to inorganic nitrites under certain reaction conditions. Also, less commonly used, but something which must be considered if they are used, are nitrosyl halides, and then there are various oxides of nitrogen that have to be considered as well. So in summary, in terms of the API process, any synthetic pathway that involves amines or amides must be considered a risk if there are nitrites or nitrite-containing reagents in the same step of the pathway. If the steps containing these two risk factors, the amines and the nitrites, are separated, then clearly the risk is going to be much lower. However, that depends on the level of separation, so that if there is a risk of carryover, that also has to be taken into account. As mentioned here, if the amines are present in a step immediately before or immediately after a step that involves nitrites, then clearly there is a risk factor involved. Now, these risks may also be mitigated in that there may be washing steps or other steps in the API manufacturing process that will remove nitrosamines if they are formed. For example, NDMA is readily soluble in water, so if there are washing steps, these can be considered a risk mitigation factor in the overall risk assessment. So now we come to Module 2. Module 2, having looked at the process risks associated with API manufacture, we now have to consider the event risks, the other risks that are associated with API manufacture. And clearly, when we talk about the risk of an event, we're talking about the risk of contamination. And this will most probably bring nitrosamines directly into the API carried on some contaminating agent. And there are three major ways by which contamination of an API with nitrosamines could happen. These are water, solvents, and crossover between processes. So let's consider those factors individually. First of all, water. There are various guidelines in terms of the quality of potable water. Potable water is typically used certainly in the early stages of API manufacture. And the internationally recognized document for water quality is the WHO guideline of 2017. And this contains a limit on nitrosamines of 100 parts per billion. There's a second document which comes from earlier. It's a 2008 document, and it explains the limit of 100 parts per billion. Now, this is the widely used international guideline on water quality, and individual countries or individual states may have their own guidelines. For example, California has a reporting threshold of 10 parts per billion in water. So it is important that the marketing authorization holder carrying out the risk assessment in relation to nitrosamines does consider the risk that there could be contamination from the water. And they should review the standards that apply to the potable water used in the API manufacture. So, for example, they should be looking at local standards as well as the WHO standard. In practice, and as has been shown by the work done by WHO, in most countries, the actual levels of nitrosamines in water are below the detection limit. But nevertheless, this factor must be considered when evaluating the risks of nitrosamines in APIs. Then we come to solvents. And when we look at solvents, we have to consider both recovered solvents and fresh solvents. It is common practice among API manufacturers to recover solvents that are used in the manufacturing process. And this is for obvious reasons, both cost and also environmental considerations. It would be impractical to dump solvents that have only been used once. There are several processes for solvent recovery. Commonly used is distillation, but there are other processes as well. And the risk that nitrosamines could make it through the recovery process has to be considered. For example, NDMA is quite volatile, and depending on the conditions for recovery, including distillation, it could also make it through the recovery process. So when looking at recovered solvents, if solvents are taken from a high-risk process, or even a single step in a high-risk process, that is a step where both amines and nitrites are present, then if those recovered solvents are used in a low-risk process, that is a process that doesn't involve amines or nitrites, the risk of cross-contamination through the recovered solvents has to be considered. So if the API manufacturer is using recovered solvents, they should consider appropriate controls on those solvents. And this includes restrictions on use. So for example, it could be that recovered solvents from high-risk processes only go back into those same processes, or solvents recovered from the manufacture of one API only go back into the manufacture of that same API. So when we talk about the recovered solvents, we should also be looking if they are released for general use in particular. There must be a testing and release process for those recovered solvents. And that testing and release process should not just consider the standard tests for purity of the solvent, but must also consider the risk of the presence of nitrosamines, in particular if those solvents are coming from any process that has a high risk of generating nitrosamines during the API manufacture. So testing and release processes for solvents must consider potentially the testing for nitrosamines. So it may be possible to recover solvents from a high-risk process using a validated recovery process, but that process has to be validated, and typically it would be validated by spiking with nitrosamine and demonstrating that the recovery process eliminates the nitrosamines from being present. The key site that is involved here is that there is an appropriate evaluation of the risk of the nitrosamines being present in the recovered solvents, and that appropriate steps are taken to eliminate that risk. Then we come to fresh solvents, and it might well be asked, what is the risk with fresh solvents? Surely that they should be arriving at the manufacturing site clean and without any risk of nitrosamines. However, many of the companies which manufacture solvents also do solvent recovery. So you might well be asked, how do you know that your fresh solvents really are fresh? And among the ways that you might establish that, first of all, your contractual and technical agreements with the solvent supplier come into play. There's also the question, have you audited those solvent suppliers? You may also require batch certification of individual shipments. And there is also the question of testing of the solvents. All of these have to be considered as potential ways to ensure that what you are buying as fresh solvents really is fresh. In addition, with fresh solvents, you have to consider the risk of contamination during delivery. It is highly desirable that solvents are delivered in dedicated tankers, so that those tankers are not used for other solvents or other products which could potentially contaminate the solvent, including contamination with nitrosamines. If that is not possible, then it would be quite typical to include a check in the receiving documentation that there is a cleaning certificate for the tanker before it was loaded. However, this question of cleaning between loads in tankers is not as assuring as making sure that you use dedicated tankers, which would be the recommended way to eliminate the risk of contamination during delivery. So then we come to the question of cross-contamination between processes. As I've outlined, some processes are high-risk processes in that they use amines and nitrites in the same step or in subsequent steps of a process. And if there is any risk of contamination from that high-risk step into lower risk steps, then that risk has to be considered and mitigated. This includes, for example, cleaning validation of the manufacturing equipment. And it includes things like campaign strategies, which may indeed be linked to the cleaning strategy. If there is campaigning of the API manufacturer, then that campaign strategy must consider, is there a risk of buildup of nitrosamines during the campaign, so that while the risk might be low in the early batches of a campaign, it might indeed be higher in the later batches. So in summary then, in relation to APIs, the manufacturer should be able to show that the external factors, such as the water and the solvents that are used in the manufacturing process, do not present any risk in terms of the presence of nitrosamines. And then there are the internal factors, such as solvent recovery or potential cross-contamination. It's required to address all of these factors in order to present a valid risk assessment. Next, we come to drug product and packaging risks. So the drug product, first of all, the major risks in the manufacture of the bulk drug product that have to be considered here are, first of all, the question of, again, of water, and secondly, the question of interaction of the drug API with the excipient matrix. So let's take those one by one. As mentioned previously, water, the potable water that is used in manufacturing, if it is used, is a potential contaminant. In general, potable water is not used in drug product manufacture. Even in solid dose products, it is typical to use purified water. The purified water purification process, in general, will remove any nitrosamines because the RO and DI processes will take out organic matter. So really, in assessing any risks in association with water, the manufacturing authorization holder should be looking at the results of their conductivity testing and their total organic carbon testing on the purified water. If there is continuous testing of both of those, continuous monitoring, then the risks can be considered low. But this does require a review to establish that those risks are low. Next, we come to the interaction of the API with excipients. If the API contains amino groups, particularly secondary amines, there is a potential for interaction with nitrites in the excipient matrix. If the drug substance does not contain amino or amide groups, then that risk can generally be discounted. But it should be remembered that a great many small molecule APIs do contain amino groups. It would be unusual to have nitrites present in the excipient matrix, but depending on the product, they may be there as part of the preservative process. So again, the excipient matrix has to be reviewed and the risks considered in terms of interaction between the API and the excipients. It must be remembered that we're looking at nitrosamine risks in the parts per billion range, and therefore the normal stability testing and so on that is associated with the drug product may not be sufficient to determine whether nitrosamines are present at that level. Finally, in terms of the drug product, we come to the packaging process. The packaging process, again, should be low risk, but there are two risk factors that must be considered. If there is printing, and this is particularly associated with solid dose tablet products which are blister packed. If there is printing ink on the inside of the foil, that printing ink may contain amines. Some inks that are used for printing will contain amines, though many will not. And secondly, in relation to the packaging processes, the lacquer that's used on the aluminium foil may contain nitrocellulose. The manufacturers of aluminium foil have been alerted to these risks, and while it is generally true that most modern lacquers do not use nitrocellulose, nevertheless it is the responsibility of the marketing authorization holder to make sure that that is the case. So these two risk factors, again, must be considered and evaluated. Finally, in this discussion, we come to the question of testing. If the risk assessment that has been done in relation to the API manufacture, the drug product manufacture, and the packaging processes, unless it eliminates any risk of the presence of nitrosamines, then the question of testing has to be considered. And the strategy for testing must also take into account that we are looking at the parts per billion range. So the testing of nitrosamines has to make allowance for having suitable facilities and qualified equipment. There has to be suitable methods. The personnel have to be trained, and there have to be the appropriate controls on reagents and standards. So let's look at each of those in turn. When we talk about suitable facilities, the organization of the facility has to take into account the risk of cross-contamination. For example, reference standards for nitrosamines should not be handled in the same area as the test samples. It should even be considered whether there should be particular gowning arrangements in terms of movement of analysts between the area where the reference standards are handled and the area where they are doing the sample preparation. The risk of cross-contamination between reference standards and samples is high in relation to, as I say, where we're looking at an extremely sensitive test going down into the parts per billion range. And indeed, the design and operation of the facilities has to consider analyst safety. The nitrosamines are listed as suspect carcinogens, so that ventilation systems and the personal protective equipment and so on must be appropriate to avoiding any health risk to the analysts. Then we come to the question of equipment. The normal analytical equipment that is typically used for assay testing, standard impurity testing, etc., is not sensitive enough to detect nitrosamines at the levels that we're talking about here. Only GCMS and LC-MS equipment are suitable for this work. And it must be remembered that that equipment must be qualified. There is some guidance in terms of the qualification of equipment in the USP Chapter 736, which gives guidance in terms of establishing mass accuracy and mass resolution. It would be extremely disappointing if there were nitrosamines present, but you missed them because of failures in mass accuracy or mass resolution. Also, there must be the appropriate checks and maintenance procedures. Modern LC-MS equipment is supplied with a tuning fluid which maintains the mass accuracy and mass resolution. But again, there must be things like maintenance procedures and checks as regards to the use of that tuning fluid. Then we come to analytical methods. Within Europe, EDQM has not yet published an analytical method for the measurement of nitrosamines in medicinal products. Individual agencies, other agencies besides EDQM and EMA, have published methods. So one of the early ones that was published was a Swissmedic method, and I've given the link here to where that method can be found. This method has a limit of detection of 30 parts per billion. And it is one of the critical factors that has to be taken into account in choosing a method. What is the limit of detection, and where is that limit of detection in relation to the levels that you would need to get to demonstrate that your product is safe? FDA has published a number of methods, and one of their methods, for example, has a limit of detection of 5 parts per billion. It is specific to sartans, so the levels of detection, or the limit of detection if that method was adapted for other products, might not be the same. And they have also established a method for ranitidine. Other products would require either a new method or adapting these methods to be used for the product in question. And if there is one of these methods used directly, then a limited validation will be required. But if the method is significantly adapted, or if it's being used for a completely different type of product, then a significant level of validation will be required to demonstrate the suitability of the method for use. Next, then, we come to the question of personnel. The personnel have to be trained in the use of the equipment and the execution of the test method, and those training records must be available. There must be assessment processes in place for the personnel before executing tests on product. So if the approach is to develop a method specific to a given product, then the transfer of that method into a laboratory that would be carrying out routine testing has to be done under protocol with appropriate reports of the tech transfer and so on. Next, we come to the question of reference standards and reagents. There are reference standards available for the nitrosamines, the 6 that are listed. But it is also permissible to prepare reference standards in the laboratory. While the reference standards are available from, for example, from USP, they are not in large-scale supply. So some companies and some laboratories may choose to prepare their own. And those reference standards then would have to go through appropriate certification and so on. Also, the reagents that are used must be free of incidental contamination. The entire question of testing for nitrosamines does involve a certain amount of risk in relation to false positives. So it is very, very important that you eliminate the risk of incidental contamination. I mentioned the risk of incidental contamination in relation to segregation of the reference standards from the test samples and so on, but the reagents used must also be considered in eliminating that risk. So finally, and in summary, If there is a risk of nitrosamines being present due to any of those factors that I've outlined, whether it's the process risks associated with the API or whether there's a risk of incidental contamination, then some testing is appropriate. It is incumbent on the marketing authorization holder to show that there is no risk in terms of meeting the thresholds that have been established by EMA. Also, if the risk is not known, in evaluating many of the topics that I've outlined, the answers might not be readily available. So, for example, the question of water quality or the question of solvent quality, it might not be possible to establish exactly that there is no risk. And if there is a significant level of unknowns, then testing has to be considered as well. And if the risks become significant, then that testing is not just a kind of screening program for the product, but it may become a regular program of testing for that product. So really it comes down to the establishment of the risk and developing a testing strategy, which may range, as I say, all the way from no testing or very limited testing right through to testing every batch. That is the responsibility of the marketing authorization holder to determine. So finally, if there's any additional information needed, I've given some directions here. You can contact NSF on this topic, and we would be happy to advise you in terms of the risk evaluation that the marketing authorization holders have to carry out, both in terms of executing that risk evaluation or in terms of auditing that risk evaluation as to whether it is satisfactory and whether the conclusions and strategy for ongoing actions are valid. So if there are questions, please feel free to contact NSF at the email address shown here on the slide. Finally, I'd just like to thank you for listening. I hope that this webinar has been of some benefit to you, and maybe we will be talking to you in the future. Goodbye for now.